for the last symposium of the official Cutting AG, beside the satellite tomorrow. Please attend. It's a great satellite. And it's about, it's about celebrating first friendship between France and Italy, our oldest ally, I'm sure. And it's about rhythms and oscillations, mostly, to make it. So why? Because they are everywhere. You can't escape them. In all spaces, they are multiple ubiquitous. They have different frequencies. They have different shapes. Some people even call them beta bursts because they want to be interesting, to look cool. But anyway, so they really, you can't escape them. It's a kind of the shining of neuroscience. And they have been under the microscope for decades. And still, well, it's not so clear what is their role and even how they are generated. So they are really widely observed, but we have only little models about how they are generated yet, and also about the computational role. And the thing is, we know that they are there. We know that they have an impact on processing. There is still this question about whether they are used by the brain. Let's say the brain could not do some computation it does without them, or whether it's just in the system because of the wiring. So at the end of this symposium, you won't have the answer for sure. You will probably be more confused than ever. But let's try to still discuss what some, let's say, recent results different groups have with different approaches. So there are several questions that are going to be addressed during this symposium. Well, let's say the origin of oscillations, more how they are generated using models, computational roles, trying to link them to some computational quantities, let's say. Pathology, also how they are maybe altered in some pathology, how they are related to feed-forward and feedback processes. So it's related somehow to computational role, and how the different frequencies interact or are dissociated with it in different networks, and how it's related to anatomy, and how it is also in different cortical layers. So it's quite broad. And the approaches will be from, let's say, in silico neural networks, it includes more modeling work, to in vivo neural network, including so EEG studies, non-human Hickok studies, human MEG, and fMRI EEG combined. So again, quite a broad view of what we can do to study oscillations and rhythms and fluctuations, I call them now, because we are changing a bit our view on it, but that's not the point of today. So let's start with Matteo, who's going to discuss mostly about this origin of oscillation, the frequent interactions with neural networks. I'm just going to read to be sure. So Matteo is a physicist, theoretical physicist by training, actually. But then he did his PhD in Parma on the interactions between structure and dynamics of spiking neural networks with synaptic plasticity. Afterwards, he did a postdoc between Indianapolis and Paris before, and that's very lucky for us and for students at the University of Lyon, joining Lyon with a junior position chair at the University of Lyon here. So his main contributions are in the field of the development of biological realistic neural mass models for cortical neural networks, and in the circuit mechanisms behind the emergence, as I said, of slow and fast oscillations in spiking neural networks, and as we're going to see today, this talk. So thank you, Matteo, for being here, and the floor is you.
Yes. Let me first check. Okay, so first of all, let me thank you, Mathilde, for the kind introduction and for inviting me to present my work in this nice workshop. So I will try to answer the question about the origin of oscillations by using modeling approaches. And in particular, I will try to dig into the question about the complexity of oscillations that today we are able to record and characterize thanks to refined experimental recordings. So this is the simple outline of my presentation. So first of all, I will start by talking about general oscillations in neural networks and how they have been modeled so far in the literature. So I will talk about the classical mechanisms for oscillations that have been studied since the 70s since many years. And then I will focus on one specific example on which we are studying, which is gamma oscillations in the hippocampus, where we study these oscillations in terms of their complexity and their burstiness. So going beyond the classical trial average analysis that we do in often. And so I will show you how this complexity emerges from a completely different mechanisms for generating oscillations, where we are not in the classical synchronized regime, but we are close to it in a region that is close to the fringe of synchronies. And then, of course, I will talk about the origin of gamma bursts. So oscillations, I don't need to explain to you, because I guess that you know very well. But anyway, oscillations, what I want to point is that are phenomena that emerge at the mesoscopic scale. The mesoscopic scale is the scale of a population of neurons. So it's typically measured with mesoscopic recordings, just like local field potentials, that measure the average activity of hundreds of neurons. So typically oscillations have been observed in different frequencies, from slower delta to faster beta and gamma, and they have been related to different computations of the brain, just like memory consolidations, rewards in addiction, and gamma oscillations, for example, in attention during spatial navigations. So there is this classical view of oscillations separated in different frequency bands. But I guess that that you already know that it's difficult to really define these frequency bands when you work with experimental data. Moreover, the origin of these oscillations, as well as their function, I will focus more on their origin now, is still unclear. There are different mechanisms that try to explain how they emerge. And one way to study how they emerge is by using models, in silico models of networks of spiking neurons, because they are the results of the average activity of neural networks. So the classical mechanism, and I will focus now on gamma oscillations, is called ping, the ping mechanism for oscillations. There is also the ing mechanism that includes only inhibitory cells, but they are related somehow. The main idea is that we have a population of excitatory cells and a population of inhibitory cells. And at some point, you can imagine that there is some excitation that activates some excitatory neurons, which in turn will activate afterwards some inhibitory neurons. And then, after some time, this will create some inhibition. As you know, this will activate conductances, and these inhibitory conductances will take some time to decay. This time constant of the decay of these conductances, things, for example, to GABA-A conductances, allows for the fact that when the inhibition goes down sufficiently, there is the possibility to generate a new volley of excitation, because neurons will be excitable. And so this time scale of synaptic conductances somehow is responsible for the period between two volleys, so for the frequencies of these oscillations. So this synaptic time constant somehow freezes the frequency of these oscillations. And in particular, in many models, it has been shown that you can have this mechanism where you have volleys of excitatory cells. Unfortunately, here they are in red, while here in blue, and afterwards volleys of inhibitory cells. And the frequencies of this oscillation is strongly related to the time scale of GABA-A conductances, and altogether, this results in gamma oscillations at frequencies from 40 to 100 Hz around. So we have these spiking neural networks, that's the raster plot of the network, and this is the global field where you observe these clean and very periodic oscillations. But our oscillations, here a little spoiler, these oscillations in real experimental data are not typically like this. So this is one mechanism, and in the hippocampus, this has been largely employed. You know that in the hippocampus, you can record the local field potentials that shows these type of traces. This is just one example where you have slower theta oscillations around 10 Hz in rodents, and on top of it, you have faster gamma oscillations. So what is typically done to make sense of this very, how to say, more or less noisy signal is to average these cycles. If you average this signal across many slower cycles, slower theta cycles, you find the very famous picture of that you observe here, where you have the color is the power of the frequency, and on the y-axis there is frequency, and here there is time. That's the average theta cycles, and what you observe very well the emergence of a slow gamma oscillations power at a specific phase of the theta cycle, and a high gamma or medium gamma power at a different phase of the theta cycles. This is very nice, and it has been shown that, for example, place cells codes differently space and time during slow and fast gamma oscillations. So in a certain sense, these slow and fast gamma, these sub-bands that you develop, that you discover here, are important also for the computations and the encoding that is done in the hippocampus. But again, this, remember, it's just an average across thousands of cycles. So by averaging, somehow, unfortunately, we throw away a lot of information. Still, we get information because we can get this nice picture, but we throw away some of the information. From the point of view of modeling, if you wanted to model this kind of situation, now you know already that you can just do this with two different populations of neurons, one with a slow decay of synaptic interaction and one with a faster decay. As the time scale of interaction determines the frequency of oscillations, you can have two different populations of neurons with identical neurons with different time scales, a slow decay and a fast decay, and you can very well model these phenomena of slow and fast gamma oscillations. And this has been done so far. And it has been, this works very well when you model the average oscillation. In a recent work that is published here, what we have shown is that the ping mechanism based on the synaptic time scale is not the only possible mechanism for oscillations. But there is an additional mechanism that emerges when you consider a crucial feature that you have in real networks, cortical networks, which is the balance between excitation and inhibition. So whenever you consider a balanced network, somehow you have this sparseness of activity of neurons and this endogenous noise in the network that somehow, as we have shown, is responsible for the activation of cycles of activity, of oscillations. In particular, by using this sparse network, what we observe is that the amplitude of oscillations goes to zero in the unbalanced regime whenever the coefficient of variation of neurons, which is a measure of their irregularity, goes to zero, as in the unbalanced regime. So whenever neurons at the cellular scale are irregular and chaotic, is where you obtain oscillations at the population scale, which is a bit counterintuitive. But that's how it is, because in a certain sense, this is not noise. This is a self-organization properties of the network. And as a result, the main difference from the previous approaches is that the frequency of these oscillations is not frozen by the synaptic timescale parameters, but you can change the excitability of neurons with an external parameter and continuously change the frequency of collective oscillations. So with this mechanism, you could model in principle not only gamma, but also beta and slower rhythms. This is one mechanism. Maybe the two mechanisms are working together, but that's just another possible mechanism that takes into account a crucial feature in the brain, which is this balance between excitation and inhibition, and the fact that neurons have this chaotic irregular activity. So after this introduction, let's go to the experimental data that we worked on. These have been recorded by Vincent Duchamp and Romain Goutagny in Strasbourg. And this work is done also in collaboration with Damien Battaglia, now in Strasbourg. So the data is recordings in the CA1 region of the hippocampus during this ecological navigation task. So the mouse needs to search for the reward, and every day, every time, at every trial, it starts from a different location. That's the local field potentials that we generate, and you can see that it is composed by many different components at different frequencies. But if you don't do this classical somehow frequency bands activity, you can study the signal without doing any frequency bands and without doing any theta cycle averages. And what you observe, which is very interesting, is that in time, you do not have only slow and fast gammas, but you have everything inside. So you have events of very powerful oscillations at 8 years, events of very little powerful oscillations at 150, 70, etc. So at every cycle, you have not only slow and fast gamma. So there is information here. So what you can do is that you can do, for the moment, very simplified methods, just thresholding mechanism methods to find these different, let's call them gamma bars. And you can estimate for each one of them their power, how important they are, their phase with respect to the theta cycle, and their phase with respect to the theta cycle, and also their frequency, the frequency where they emerge. And if you plot this in this three-dimensional plot, you observe that across all the cycles, you have many frequencies, many amplitudes, and many different phases. So one could think that actually these little events are just noise. That's a possibility. What I want to say is that then if you take all these events and you average across the theta cycle, you obtain back the classical view that I showed you before, where you have slow gamma oscillations and fast gamma oscillations across all these cycles. But still, inside all these time traces, you have all this huge variability of the events. And what we have done is to answer the question, are these events just noise? So to do this, and this has been done with Damien Battaglia, who is an expert in this kind of analysis, we have trained a classifier, and we have tried to decode the position of the mouse in the ecological space based on the features of the gamma events. And in this case, the features are simple, which are only this one, which are the frequency, the phase with respect to theta, and the power. Of course, you can decode the position of the animal, but what is more interesting is that even when you consider only those gamma events which have a very little power, which could be like noisy, like these ones, okay, you still can decode the position of the mouse. So this means that these events are not only noise. They have a code inside, and they bring information about the behavior of the mouse. So this tells you that making the average gives you a nice representation somehow. You can do the average, and it gives you important information. But if you don't do the average, you can keep track of a much richness of events that still bring information. So this is very important for us. And what we were then asking ourselves is, how do these events with different frequencies and phases and amplitudes emerge in sparking neural networks? Are they a natural thing to expect? Which doesn't seem to be the case, because in all the previous models that I have shown you, also the one that we have studied, oscillations are typically like this. So they are very sustained, like very clean, like a limit cycle in terms of dynamical system theory, and which works good for the average oscillation somehow, but not if you want to model this trial by trial variability, or more specifically in this case, cycle by cycle variability. So in order to do this, what we have done is that we have employed our mechanisms for oscillations for balanced network, and we have employed excitatory inhibitory neurons, so a much more realistic network. Before it was only inhibitory neurons, and we have given an external excitatory theta drive to our networks, like we have in the hippocampus. And what we have done with this simulation of these spiky neural networks, we have measured the amplitude of the oscillations in function of parameters of the model. The two parameters is the connectivity of the network, so how many connections neurons do on average, and the coupling from inhibitory cells to excitatory cells. So what you observe is that increasing the connectivity, we have an increase of the amplitude of oscillations. In particular, you can define a line of transition from two different regimes that has been traditionally called the asynchronous regime, where you don't have oscillations, and the collective oscillations or synchronous regime, where you have the classical sustained oscillations. So what we have done is to say, let's look in all these regions, which is at the fringe of synchrony, so close to this critical point. Remember that we don't need to go very close. We could stay in all this fringe of synchronous region. So let's take one point in this asynchronous region and let's check the dynamics of the network. If you do this, you find you can model a proxy of local field potential signal, and that you can analyze with the same methods that we did for experimental data. And what you observe in this spiky neural network is that naturally it emerges not clean limit cycles, but gamma bursts at different frequencies and phases with respect to the theta cycle. And if you do the representation of these events in terms of their amplitude in power, the frequency, and the phase, we observe a very similar pattern of events like those that you observe in the data. So in a certain sense, in this mechanism, somehow we need to be at the what we call the fringe of synchrony to model this complexity. So the idea behind this is that somehow being close to this critical point allows for the network to explore different little attractors of possibly coordinated subgroups of excitatory and inhibitory neurons that are excited just by the fact that in the system there is endogenous noise because of the balance between excitation and inhibition. And so from time to time, you have these bursts of event. So remember, that's important for me. In the model, these bursting events are not noise. In the model, there is no source of noise. The model is completely deterministic. Once you start the simulation, you can predict everything that will happen in the future. These events are in this case, the result of their wiring of connections. And so they are not noise. Okay, so this is important. So then we studied some further features of these dynamics, and in particular, we studied the entropy of the power spectrum in function of always the connectivity and the inhibitory couplings, and the spike train entropy, so how neurons are chaotic. And we observe, as we have already somehow said, is that in all this region, that is what we call, remember, the fringe of synchrony, we have a high entropy of the power spectrum, and at the same time, we have a high entropy of the spike trains. So this region is characterized, because of balance, by extremely chaotic activity of neurons, which doesn't mean noisy and completely unpredictable. This means just that is irregular as it should be in the brain during balance sparking dynamics. But what about the beam burst? Can we change something about the network, or we cannot avoid this complexity? Well, we can estimate from all these events the distribution probability of gamma events that you find here. And for example, for this set of parameters, you observe that you have this distribution of events is peaked at 40 Hz. And if then you change the amount of inhibition, so the strength of GABA A conductances, you can shift the network to being mainly slow gamma oscillations dominated, to being somehow dominated by, at the same time, slow and gamma oscillations with the same power, and to being dominated for large inhibition by fast gamma oscillations. So what I want to say is that this reconciles with the idea somehow of frequency bands on average. But inside each of these simulations, we have all these large complexity of events that we observe also in the data that we wanted to take into account. Okay, so now just a few words about some works in progress that for me are interesting. So we want to start to study how these events are somehow related to the sparking activity of the neurons of my networks. And so we have done some very simple things, like, for example, in this case, we have tried to decode if neurons are active or not based on the features of the gamma events. I have to say that the result is not very exciting, because we could decode the neurons are active based on the different gamma features for only few neurons, in particular for only 12% of the neurons. But still, for these 12% of the neurons, we could decode based on the gamma features if they were active or not during the gamma event. So somehow to link the sparking activity of the underlying network to the features of the gamma events. But for this analysis, it's, I would say, for the moment, very unsatisfactory. And that's maybe because our way to characterize the gamma events is too much simplified, and the model is not structured at all, so it's almost maybe too simple. So what we want to do now with some collaborators also here in Lyon is to study how different bars can be linked to the creation of different neural assemblies in the underlying sparking networks. So to give a mechanistic understanding of this phenomena. So few words about a couple of things. This is about sparking neural networks. What is typically done also in computational neuroscience is to study larger scale network. And to do this, one needs to have models at the mesoscopic scale. Population models are called, that then represents one node of this large scale whole brain model. Mean field theory is a powerful tool to develop low dimensional description of the activity of these sparking neural networks with few differential equations. So a dimensional reduction to model the population activity of this network without losing information about all the complexity of this network. So I don't want to go into the details. So it is possible to develop neural mass models, so low dimensional description of sparking neural networks for balanced network, and taking into account, for example, also the heterogeneity of excitatory and inhibitory cells in the network. So here is a result by Manoj, who has a poster here that I invite you to go and check out. So what you can do with these models is that you can really study more the phase space of the model. And you can see that based on different parameters like the external drive and the heterogeneity between neurons, so the fact that different cells have different excitability, you can find many different dynamical regimes. from asynchronous states to the classical ping to bursting oscillations like the one that I have shown you before. And what is interesting is that if you estimate the phase amplitude coupling of these oscillations with the corresponding theta cycle, you find that the bursting region, which is this one, is the one that has the maximum phase amplitude coupling, which is much stronger than the phase amplitude coupling that you would have with the classical ping and sustained limit cycle. So in a certain sense, this burstiness is also able to boost the interaction and the cross-frequency, the coupling between these fast rhythms to theta cycles. Okay. So about conclusions, I showed you I talked about the trial by trial richness of these gamma events, saying that if we do not do averages across trial, we can still have a lot of information about the dynamics of our networks and these oscillations, and that using models, we can suggest a new mechanism where these events arise at the fringe of synchronies due to the balance between excitation and inhibition at variance with previous mechanisms based on synaptic timescale of interaction. The mean event is controlled by recurrent inhibition. Here I miss an I. And again, the bursting regime somehow maximizes the phase amplitude coupling with slower rhythms. So I thank you for your attention. I need to thank also my collaborators, Vincent Duchamp and Romain Goutani, who did the experimental recordings in the hippocampus in Strasbourg. Damian Battaglia, who did a lot of analysis on the classifier for these gamma bars. Manoj Nandi and Alessandro Torcini for the theoretical development of the neural networks of underlying oscillations. And of course, I thank you and all the listeners for your time and attention.
Okay, so thank you very much all for being here. Thank you, Mathilde, for organizing this symposium. It's very, very cool to be here. And thanks to the organizer. It's very awesome to be in this garden. So today I'm going to talk indeed about traveling waves, so oscillation interpreters, traveling wave, and their relationship with predictive coding. And I will start, if I manage, yes, from actually the work of that I did during my postdoc with Rufan. So we start investigating, so he actually showed the story start in 2012, so a long time ago, where he showed that if a participant is attending to a visual stimulation with flickering over time, if you cross-correlate this white noise, this visual stimulation with the EEG, then you obtain an impulse response function that basically tells you how the brain responds to variation in the stimuli. And what we found is that this impulse response function oscillate in the alpha band, so in the around 10 Hz. So we basically adapt these perceptual echoes and start investigating how oscillations are related to perception. What we care about in my postdoc is whether we could replicate these oscillatory dynamics related to perception using predictive coding. Nope. Maybe I will use this guy. Yes, easier. So let me remind you in a nutshell what is predictive coding. So the idea in predictive coding is that basically sensory systems are thought as generative hierarchical processes, where higher region predicts the activity of lower region, and the difference between the prediction and the actual activity is called prediction error and is used to update the next prediction. So the intuition is that the brain has a model of the world that allows us to interact efficiently with the environment. And that's basically what we did. So we have a very simple model with two brain regions that could be V1, V4, or whatever. And we have our Y, which is a prediction, and our prediction error, X. So X is just the difference between the input, so the visual stimulation, and the prediction. So how wrong is my prediction in predicting the input, the difference. And the prediction is updated based on the prediction error. So that's a very fancy model to not do much, actually, because the Y is just tracking the input. So the prediction would be exactly the same as the input. So we decide to spice things up a bit, and we add biological constraint, specifically the temporal delay. So now it takes some time for the information to travel between regions. So the prediction error will be always a bit late in updating the prediction, and the prediction will always a bit late in predicting the input. And we also add temporal time constant, basically, to model the dynamics over time. And it's important to notice that the time constant of the prediction error is much faster than the time constant of the prediction. It's a detail, but it's important because you want to be fast, basically, in tracking the input, in updating your prediction, but based on the prediction error, but you want to be slow in changing your prediction. Okay, so we did this simple predictive coding with a bit of biological constraints. And what we found is that if we consider our prediction as a proxy of our EG, so we consider the Y is basically what we record with the EG, then if we cross-correlate once again the input with the prediction as we did experimentally, so again the input is just white noise, so just fluctuation over time, then we were able to find our oscillation in the 10 Hz. And here we use specifically tau of 17 milliseconds, not 15, and delta t about 12 milliseconds. So we were already quite happy because with a very, very simple model of predictive coding and biological constraints, then we are able to recreate these oscillatory dynamics. Okay, but maybe we were lucky, right? We took these two values and we got the right oscillation. Well, then we explore the parameter space. So we vary delta t and tau over 0 to 30 milliseconds, which are biologically plausible range. And we see how the oscillation change. So the color represents here the frequency of the oscillation. Red is the alpha oscillation. And we saw that for a range of 15 plus minus 5 milliseconds, more or less, we are in the alpha range, which is pretty cool. So it's actually a stable phenomenon that can replicate within a range of biologically plausible value. And another interesting observation is that if we get shorter in time, so if the interaction between brain regions become faster, so a more local phenomenon if you want, then the frequency increases. So we go toward beta, if not gamma oscillations, which is also intuitively kind of makes sense. Okay, then we get excited. So let's push this even further and let's expand the model from 2 to n layers. So it can be as long as we like. The equation looks a bit scarier, but it's exactly the same idea. So we have our prediction error, which is just the difference between the prediction and the activity of the previous layer, which when L is 1 becomes the input. And our prediction, again, is updated based on the prediction error, and on also we include a prior. So there is now some prior activity that is predicting the environment. In this case, it was the same as the input, so it was still a white noise. And it now contributes also to the change of each prediction. But the idea is exactly the same. And if we expand our network to different layer, what we found is that these first, we are still able to replicate our oscillation. So here you see on the x-axis the time, on the y-axis the layer from 1 to 7, and the color code represents the activity of each prediction, if you want. And yellow is the peak, and blue is the trough of the oscillation. And what we found is that if we feed the model with the input, so if we keep giving a white noise to this model, then the activity of our prediction oscillates and propagates forward as a traveling wave. So there is a constant phase shift between the first layer all the way up to the last layer. And if we kill the input, so we keep only the prior activity, then we observe still an oscillation, but this time propagating in the opposite direction. So start from the prior and travel top down to the lower layer. Okay, so our model kind of makes this prediction. If we have an input, then we find some alpha oscillation that propagate forward, whereas if we don't have an input, then these oscillations are still there, but propagate in the opposite direction. Okay, so we try to validate this in experimental data using EG. So how does it look like in actual data? So we took our 7 layer became the 7 electrodes in the midline. So from OZ occipital all the way up to FZ. And you can see these are already real data. And you can see already by just looking at the graph that there is a constant phase shift from occipital to frontal electrode. So this wave is kind of propagating forward. And this was in case of visual stimulation. Here is a more schematic, but maybe more intuitive way of seeing the traveling waves. If you have our activity of electrodes, so occipital and frontal, then a slow oscillation that starts at the occipital electrode and propagate forward, this is a forward wave, whereas a wave propagating in the opposite direction, so starting frontally and traveling all the way down to occipital electrode, well, that's a backward waves. Okay, cool. We defined our traveling wave, but how do we quantify them in practice? So we have our data, we can plot these nice images, but then how do we actually quantify them? When to do that, we move to the spectral domain. So let me remind you in a nutshell how the 2D FFT works. It's basically an FFT, but in 2D space, so basically for images. So if you have an image like this, where you have an oscillation that propagates from left to right, so again, white is the peak and dark is the trough of the oscillation, then if you do a 2D FFT of this image, you get a spectra where there are these two peaks, which correspond to the frequency of this oscillation along this axis. And vice versa, if you have an oscillation that propagates top-down, so you start from up and travel all the way down, then again, you do a 2D FFT and you get two peaks on this axis. Okay, nice. And of course, the peaks represent the frequency. Okay, now if we rotate a bit these images, clockwise or anti-clockwise, also the peaks will move. So now the peaks will be in this quadrant when you have this kind of pattern, and in these two quadrants, in the opposite quadrant, when you have the opposite pattern. Well, that's exactly what we need, right? Because this looks very much like our forward waves propagating from occipital, let's say, to frontal region, and vice versa. This is our backward waves propagating top down. So bingo, we have our results. We can take from this quadrant and this quadrant the amount of power for backward waves and for forward waves in each frequency. Very nice. How do we do actually in real data? Well, we have our, let's say, 6 seconds of experiment or whatever, so our signal, our N electrodes, could be 7, could be 5, and we create our images. So this is our real data. We could do our 2D FFT, okay? So we can quantify now on this quadrant the amount of power in the forward waves, like we showed before. So we are in this quadrant, forward waves, okay? And the lower quadrant represents the backward waves. Nice, so we can quantify, we have the power for each frequency, especially for the alpha, but we can do it for all the frequency, for forward waves and for backward waves. But it's yet not enough, right? Because we need a baseline. And how do we compute the baseline? Well, we do the same thing, but this time we shuffle the order of the electrodes. We do this several times, and the idea of shuffling the electrodes is to destroy the spatial information. So the temporal power is still there, but now the spatial information is mirrored across the two quadrants. So we have basically, we can quantify our baseline as a surrogate of forward waves. So this is our surrogate of forward and our surrogate of backward. And this is our baseline, basically. Okay, then we combine the two with a log ratio. So we take basically eventually everything that I will plot in the following slides. So the experimental results are expressed as the ratio, the log ratio between the power and the power on the surrogate data, our baseline. Okay, so first experimental results was work done by Cao Yang, who was a PhD student in the lab, and she basically investigated how the traveling wave propagates forward or backward during visual stimulation. So try to replicate the results of our experiment. And she had two conditions. In one case, the stimulus was still flickering like crazy, so white noise, like in the very previous slide, or it was a very boring 5 seconds white patch that stands there for 5 seconds. So 5 seconds of stimulation, and then 5 seconds of blank. Okay, and the idea was to quantify the amount of traveling wave along the midline, so from OZ to FZ, during visual stimulation and during in the absence of stimulation. Here are the results. On the left, you see the dynamic condition. On the right, the static condition. On the x-axis is time. So the shaded area is when there is visual stimulation in gray, and the white area is when there is no visual stimulation. Blue are the forward waves propagate from occipital to frontal, and red are backward waves, so in the opposite direction. And what we can see is that there is no waves basically in the, when there is no, there are no forward waves during visual stimulation, but a lot of forward waves. So alpha propagate mostly top down when there is nothing happening on the visual screen. But during visual stimulation, well, there are both of them. So there is an increase of forward waves and a decrease of backward waves, both in the dynamic and in the static condition. Interestingly, in the static condition, where it's more predictable or more boring, if you want, so it's just a white patch that stays there for 5 seconds, but over time, the forward waves decreases and the backward waves increases. Okay, so now one may wonder, okay, what's actually happening during the visual stimulation? Are they happening at the same time? Do they alternate? And what we found is what Cao Yang did was a correlation trial by trial between backward and forward waves. And what she found is a negative correlation between them. So basically, when you have backward waves, you don't have forward waves and vice versa. So they basically alternate over time. Okay, so we were quite happy because we could relate forward and backward waves to our visual stimulation. and in line with the prediction of our model. So now we go to the more recent kind of work. So that was the very starting point. So we have a nice model, very simple actually, based on predictive coding that can generate oscillation that propagate forward or backward. And so backward waves supposedly encode prediction. So that's trying to poke into the functional role of oscillation. And the forward waves supposedly are carrying prediction error. So let's push a bit more the interpretation. If we imagine that there are two layers in the brain, one has the prior representation. So this is your model of the world that generates the prediction. And the prediction are sent back with these backward waves. And then you have the sensory data somehow, so the likelihood in a Bayesian interpretation. And the difference between this prediction and the actual data, the prediction error is sent back. And these are the forward waves. So this is the normal condition. Let's say you have a model of the world. Could be whatever, the next stimuli or the distance of the car in front of you while you are driving, with some uncertainty. So you think the car in front of you, it's about 3 meters, but plus minus something, depending on the condition. And in some case, you will have a better prior, so your precision will be much higher because you are really sure about the position of the car, for example, because it's a very clear day and you are driving slowly and everything is fine. So you are very sure it's, okay, 3 meters plus minus 20 centimeters. I'm quite sure about that. And if we are in this case, well, supposedly there should be more prediction going top down, so more backward waves and less bottom-up waves. Or in the other case, it's a very foggy day or it's raining as today, you have no really idea where the car is in front of you, 3 meters plus minus 2. And so you actually have less precision in your prior. So your representation of the world is a bit more messed up. And in this case, you have less strong prediction and much stronger prediction error. So more bottom-up waves. And that's basically one of the main hypotheses that I like to test experimentally. So we have some data in which we try to do this in a kind of undirected way. So the first approach is in a work that has been done in collaboration with Robin Card-Hartis and Christopher Timmerman. And these two gentlemen in London, well, now Robin is actually in California, but they used to be in London, and they work in the Psychedelic Center. And they studied the effect of psychedelic drugs on subjects, basically. And what they did, they give DMT, which is a very powerful psychedelic drug, a serotonergic drug, which is the one used in, for example, in ayahuasca ritual, in shamanic ritual. So it gives you a very powerful conscious, altered conscious experience and elicit visual stimulation, visual hallucination. And they have a prediction, Robin and Carl Friston, they basically suggest that psychedelics, what they do, they basically messed up your prior. So when you are under psychedelics, so if you have DMT or psilocybin or any of these psychedelics, what happens is basically they messed up your representation of the world. So they smear up your prior. And if that's true, if their hypothesis is true, then if we record EEG data after DMT, then we should see an increase of bottom-up wave and a decrease of top-down waves. So they kindly share data with us, EEG data, where participants were given placebo and DMT in different sessions. And here are the results. So the dashed line is the placebo, pre and post injection, and participants were having eyes closed during the whole both sessions. And so what you see is that we basically here replicate once again the result we had before during placebo. So no forward waves before and after, well, it's placebo, not surprising, and a lot of backward waves. So, okay, that's nice. It kind of replicates our results. Now let's go to the DMT, which is the interesting one. Before DMT, like placebo, no forward wave, lot of backward wave. But surprisingly and interestingly, after DMT, there is a surge, a fast increase of forward waves and a nice decrease of backward waves, very much in line with the prediction of the model. So basically, there is an increase of prediction error if we stay in the predictive coding interpretation, and a decrease of prediction. So this matches the prediction of our model, and it's very interesting to compare the time course of DMT and visual stimulation. Of course, it's different time scale, and of course, the comparison is quite speculative, but it really looks similar. So it's tempting to think that DMT pokes in the same neural mechanisms that are involved in visual stimulation, also given that it elicits visual hallucinations. Okay, so nice. we could say that both forward and backward waves are influenced by psychedelic drugs. And supposedly, I would like to interpret this again in the light of predictive coding. So forward waves are representing more prediction errors. So DMT is actually mirroring your priors. So we are in this case, more forward waves and less top-down waves. Very cool. Okay, first prediction kind of verified. Now we go in the opposite direction. Okay, how can we test this? And again, we had this another collaboration with Dario Gordillo and Michael Herzog in Lausanne in EPFL, and they work on schizophrenia patients, among other things. And there is one idea, one hypothesis that suggests that in schizophrenia, one of the interpretation is that schizophrenic patients, they have tighter or more precise prior. They rely much more on their prior than on sensory data. So we are once again on this case. So the hypothesis would be that if we compare traveling wave in a schizophrenic patient and in healthy control, there we should see more top-down waves in the schizophrenic population than in the healthy control, and less forward waves in the alpha band. So we collaborate together. They share kindly again their data, and what we found. So we test. Here I should have shown little by little, but sorry, just let's go step by step. So we had 121 patient and 75 control who spend 5 minutes or 10 minutes with the resting state, so closed eyes. On the left, we have the power for each electrode. So this is the 1 DFFT, no traveling wave, just the raw power. On solid line is the control for each frequency band, alpha, theta, low beta, and high beta, and gamma. And for each electrodes. And so overall, there is more power in alpha and theta, for example, in the patient. But what we actually care about is the traveling wave, right? So we perform our traveling wave analysis in decibel for all the spectra. So here you see all the spectra from theta, alpha, all the frequency band. And here is the average of the frequency band as highlighted here in different colors. So we focus on theta, alpha. We split low beta and high beta kind of arbitrarily, and gamma. And you can see here. What we care about for our hypothesis specifically is alpha, right? So our hypothesis was that a patient, they should have less forward waves and more top-down waves because they rely more on their prior. And that's what we found. So here it's a bit easier to see. In the alpha band, we have more waves in the patient population than in the control, and in the top-down condition, and the opposite case in the forward condition. So more waves in the control than in the patient. So again, we were quite happy because we kind of validate both prediction on both sides, still kind of indirectly, but we kind of managed to, again, match the prediction of our interpretation of our model. Another data set that we have is the same patient. They did the 10 minutes of resting state, but they also did a vernier visual task in which they had to judge whether two lines were a line on the right or on the left, as here. There were 4 conditions. I will not enter into details because there are basically no difference between the 4 conditions. But the idea was to investigate whether a visual stimulation, a visual task, whether there were difference between patient and control in a visual task, basically. So in this vernier visual task. So again, same number of control and population and patient, and here same results. So 1 DFFT, so the power for each electrode that we investigate from Z to FZ for the 2 population, and overall there is more power in the patient as in the previous data set. For the traveling wave, this time we find kind of surprising results, in the sense that for the forward wave, for the backward wave, we still see overall that schizophrenic patient, they have more power everywhere in all frequency band, including alpha, but it's a broadband phenomenon. But it's also true for the forward waves, not for theta, but for all the other bands. So it's a bit harder in this case to interpret these results, also because the task honestly was not really designed to test a predictive coding interpretation. So we were a bit curious, but yeah, we cannot really do much or go much farther in the interpretation in the predictive coding model. And finally, we correlate the waves between dataset, just to make sure that we are actually measuring the same thing somehow. So this is the correlation for the patient group. On the y-axis, we have the visual dataset. On the x-axis, the resting state dataset for backward wave, forward waves, and each frequency band. So we find a nice correlation in basically all frequency band, except in alpha, which is supposedly because in the resting state and in the visual state, alpha is involved differently. So in the resting state, probably is more idling. In the visual, maybe is doing actual visual sampling or whatever, but supposedly there are different processes going on there. In schizophrenia, because it's not quite the case for the control. In the control, we were actually able to find a correlation pretty much everywhere, also in alpha, except in theta, which is not surprising because theta is possibly confounded by the ERP's response or due to the visual stimulation. So that's kind of expected. So it's kind of reassuring that we were able anyway to correlate between data set the amount of waves, because it kind of said we are measuring the same thing. So that's also nice. Lastly, we went a bit farther. So schizophrenia patients, they were under pharmacological drugs, so they were taking, of course, treatment, which is measured in CPZ equivalent, and we try to correlate the amount of waves in every frequency band, in every direction, with the amount of the drugs that they take, and there was no correlation whatsoever. And we also correlate that with their quantification via questionnaire of their positive and negative symptoms. That are two waves, two symptoms that characterize the schizophrenia disorder. And we didn't find, again, any correlation between waves and the positive and negative symptoms, which is kind of disappointing. Somehow I was hoping for having a diagnostic marker, but actually no, I was hoping too much. Okay, so let me wrap it all up. So the first take-home message is that it's interesting and important, I think, to consider traveling with oscillation as traveling waves. So it's cool to think about oscillation, but not only as a temporal phenomenon, but also as a spatial one. So how they propagate through the cortex. And we know that they are involved in visual stimulation, that they are affected by psychedelic drugs. And I didn't show in this presentation, we also investigate the role of both forward and backward wave in visual attention. So we know that the visual attention modulates also alpha waves, both forward and backward. And there is ongoing work that I'm currently doing to understand how traveling waves are modulated during binocular rivalry in schizophrenia, as I show you, and in working memory. And I'm trying to interpret all of these again from the point of view of predictive coding, because I really like the idea of having a framework that kind of can explain and tell us about this. And the very last thing, all the results here I know are very indirect, because in both cases, both for the DMT and for the schizophrenic patient, is very indirect, because you have to kind of, there is the assumption that DMT is altering the prior, which maybe is not true, and the interpretation that schizophrenia is in this case, which again, maybe is not true. So all these evidence are very indirect. The idea what we are doing now is try to test this experimentally directly by manipulating the prior experimentally. So thank you so much for your attention. Thanks all the collaborators that were involved in this work and my team at the Circle. Thank you very much.
Thank you. That was really cool. You mentioned a bit about the question I was going to ask in like your last slide. And that is like, what is the null model here? Right? You could imagine that if you set up a hierarchical model with temporal delays, that you will get traveling waves, and the direction of those waves will be, the direction based on the relative, strength of inputs on one end or the other. And so in schizophrenia or DMT, you're just altering activity, in visual cortex or frontal cortex. So could you get this sort of thing, regardless of what signals were passed between nodes in the network, whether it was predictions or prediction errors or something else?
Sure, I guess it would be possible to have a model that it's not strictly doing predictive coding. So it could be just some other forward or backward information, and then you alter occipital, you alter frontal, and you alter the waves. Yes, I think that would definitely be possible. I still like the predictive coding idea because it gives a theoretical framework, and we can make predictions somehow. So the idea now would be to alter prior based on expectation, or with statistical learning or mismatch negativity. So violate a prediction, so actively play on the prior, poking on the prior to make sure that it's indeed predictive coding that is happening. But I agree that it's not the only options, and that's what I like to test more directly. Yeah.
Could I just maybe go?
Can you hear me? Yes.
Yeah.
I have a question about the interpretation of this traveling of the waves. Where do you think that they're traveling? Because I always thought, but maybe this is a naive idea, that you can even have very constrained oscillatory activity that simply by changing the orientation of the activity would have been seen by these electrodes at different delays. And so this could be a wave that doesn't travel at all, stay constrained, but because of change the effective dipole orientation, in the end you see the different electrodes.
Yes, totally. So there is this work from Zigalov and Ole Jensen recently, in which they model the two dipole, and for forward waves at least they say, okay, you just need two to actually observe the pattern of forward waves. Cool, awesome. That makes sense. And it still kind of could be in line with predictive coding or not. I definitely don't think that is really all electrodes, each area having is activity and doing is much richer than that probably. I'm leaning now toward a cortical thalamic thingy. So there is the pulvinar actually that play a role in getting the information and send it back. And I'm trying to interpret this in the light of predictive coding. And there is a paper from the 80s from Mumford who already get it all. He thought that the pulvinar as a blackboard theory, as a blackboard basically, that get information from the cortex, integrate it, and send it back to the cortex. And I like the idea that maybe the delay and the way the pulvinar is doing this integration is what generates the pattern of oscillation. So yes, I totally agree, and I don't think it's actually each area doing the predictive coding thing. It's a bit too simplistic. But I also think that there is more than just dipoles. I mean, that's what I like to test. Could be that it's just two dipoles, and okay, I would be a bit sad, honestly. I would like to have it a bit richer, but I like to test it more explicitly, more directly, and maybe also with intracortical recording as well. That's something that I'm planning to do in collaboration with Leila Reddy at Cerco.
Another question. Can you do the same with
Ooh, proprioception. I don't know. We was thinking about auditory or other kind of modality, but it's a bit harder to know how the waves would propagate. For vision, it's easy. You have the ventral and dorsal. It goes this way, easy peasy. With the EEG for the auditory, it's trickier to know in which direction. We thought of it a lot, and we were thinking to do multimodal integration. That would be very cool also to interpret in a Bayesian sense, but I'm not sure I would know with EEG how to do that reliably. But that would be very cool, yeah.
Thank you very much for a very interesting talk. I'm wondering whether you try to look at the point where actually these forward and backward waves like meet each other, or what happens when all these two like waves. Yes, and I'm wondering, do you think it could be somehow related to the fact that alpha oscillations dominate in the occipital regions in the human brain?
Right, so I'm not sure they actually meet. So what Tsai-Yang showed with the negative correlation is that they alternate each other. So there is one or the other. So they never happen at the same time. If they happen at the same time, I guess they would overlap and you would see that as a standing wave, I guess. Sure. Yeah, I don't know. I wouldn't know what would happen. I guess they would cancel each other out. I guess depends on their phase, and you would see either a standing wave or you wouldn't see much because they cancel each other out. But that's not exactly what we see in the data. But it's a good point. That's yeah, I don't know exactly what would happen in the model. Everything happened at the same time, of course. And because of the difference in the tau, so basically what you were suggesting, that depending on how much weight you give to the input or the prior, then you see one or the other. So in this case, the input takes over because you want to see what's happening in the world. You want to be fast in reacting to the environment, and you care a bit less about the prior. So maybe there is a specific phase in which you, okay, let's now see what I'm expecting, and then I actually update my expectation based on the environment. So yes, but so yeah, I actually don't know. That's a good question. Yeah.
Any other question? I have one actually. Well, the experts on predictive coding are not here, so maybe we should talk to them afterwards, because also on the effect of precision on prediction error, I had the feeling that it was the opposite, but more precise, leading to more prediction error when you have surprises. That's when you have. But that's not what you should. But no, my question is more about the other works regarding predictive coding, like maybe Marcus Borer had some work using indeed expectations. I mean, they had some expectations, and then you have surprise, all this manipulation. And what they observed was more like, at least they looked only at power, not traveling wave or anything. It was an imaging, and it was more relating gamma power changes to prediction errors. And I think they are overworked doing that. So I was just wondering whether indeed, as you said, like, are they really related to prediction and prediction error, or are they carriers mostly, and possibly gamma would be nested in that, and that.
Totally. So for the first point, in this case, I'm talking about the precision of the prior. I think what you meant is the precision of the prediction error, because all you can imagine the prediction error also to have a precision.
But still, I mean, but let's talk about the specialists, because
for the second point, yes, totally. I think that alpha is definitely, I mean, the way I see it is more like a carrier indeed and gamma is doing the actual heavy lifting job locally. And there is also the work of a lot of work of Bastos, or that actually relate gamma to prediction error. And the idea would be, one of the idea that we are also working modeling is to have a multiplexing mechanism where the alpha is actually carrying around information and gamma is doing, gamma and beta in the cortical in different laminae are doing actually the job of computing the prediction and prediction error locally. So yes, I'm definitely going in that direction. Okay,
so you agree that alpha itself does not carry information. I mean, it's not. I mean, it does carry information.
Right, but not. Yeah.
Okay.
Yes.
Okay, good. Well, thank you very much.